Welcome to the Department of Medicine Grand Rounds. Today's talk is part of the Physician Scientist in Medicine series. Our speaker is Dr. Hana El-Sali, who is a professor in the Departments of Molecular Virology and Microbiology, as well as the Department of Medicine at Baylor College of Medicine. Dr. El-Sali received her medical degree from American University of Beirut, did internal medicine residency at the University of Connecticut, followed by a fellowship in infectious diseases at Baylor. She stayed at Baylor, she joined the faculty, rising through the ranks to her current position of professor with tenure. Dr. El-Sali is an international leader in translational research on vaccine development. Her many publications include research on vaccines for a broad range of diseases, including, but not limited to, influenza, anthrax, tularemia, malaria, parvovirus infection, and pneumococcal disease. She has made seminal contributions to the study of SARS-CoV-2 vaccine and therapeutics development. She's a national co-principal investigator and co-first author of the NIH-sponsored study that established the efficacy of the Moderna mRNA vaccine to prevent COVID-19. She's the Baylor principal investigator of the NIH-sponsored adaptive COVID-19 treatment trial, or ACT studies, that generated data culminating in the licensing of remdesivir and in the emergency use authorization of baricitinib for the treatment of hospitalized patients with COVID-19. In addition, Dr. El-Sali leads a large portfolio of federally funded research as principal investigator of the NIH-sponsored Vaccine and Treatment Evaluation Unit, or VTEU at Baylor, and oversees a host of active funded trials through that unit. She has received a number of honors and recognitions for her work. Most recently, the prestigious Michael E. DeBakey Excellence in Research Award at Baylor College of Medicine. We're truly delighted to have Dr. El-Sali as today's Grand Round speaker. Dr. El-Sali, the floor is yours.
Thank you, Rolando, for this kind introduction, and thank you all for joining this afternoon. Today, I will be going over my journey as a physician scientist here at Baylor College of Medicine, with a focus on one disease entity, which is COVID-19, and how we worked here to help provide vaccine and therapeutics to stem the pandemic. These are the learning objectives. And importantly, I need to be able to answer Rolando's questions regarding what made me a physician scientist and kept me one, and how my clinical practice and research interact. So to start from the beginning, I started in a whole other disease and a whole other epidemic. After fellowship, or late in my fellowship, I joined a group that was here at Baylor, heavily invested at the time in TB research. And it was a large 9-year population-based active surveillance and molecular epidemiology study of TB in the county. And I joined in my last year of fellowship and stayed on in my appointment here at Baylor as junior faculty with the group led by Drs. Musser and Gravis, who are now at the Methodist. This particular large research project was really a good training ground for understanding epidemiology, statistical tools with the lead biostatistician Larry Teeter at the time, medical writing, and basic lab techniques pertaining to MTB genotyping, PCRs, and ELISAs. I did everything really that would have been a good K grant award, a K grant submission, I should say, but I was not eligible for it because I was a physician on a visa. But lesson number one, there is more than one way to skin a cat. okay, probably is not the only way to go about it. We were very productive in that group because, you know, it was a good team. Everybody was passionate about what they were doing, and we made numerous contributions to the epidemiology of TB study. My first grant submission was an R1 that was not funded. It was in the MAC, actually, epidemiology. But the second one did, and it also was pertaining to TB, moxifloxacin drug resistance evaluation. We looked at all the clinical isolates from Houston and their genotype and susceptibility profile. At the time, in the early 2000s, quinolones were being incorporated and evaluated as TB treatment regimens, TB treatment regimen components. That was a federally funded project. It was an R03, and because it was an R03, we relied heavily on coordinating with State Public Health Lab and Houston Department of Public Health to get a lot of the data needed for the project. And we published our findings showing that the drug resistance to moxifloxacin was very low. And however, only 50% of it can be determined by the gyrase mutation. We had postulated other genes could be contributing to the drug resistance, but we did not show that they did. Shortly after I joined the group, however, despite it being quite a productive group, it lost its NIH funding. So it was time to turn the page. At the time, I was shadowing or working closely with Dr. Wendy Keitel on a project evaluating the shingles vaccines, Zostavax. I was covering her especially over the weekends because we had to evaluate each and every rash that occurred on every vaccine or placebo recipient on the study, and there was 1,500 of them. So it was a rather involved project, but it was kind of on the side. But the group within which this study was occurring is rather an august group here at Baylor. They have a multi-decade history in evaluating vaccines and therapeutics, leading to licensing and marketing of various vaccines. Listed here on the slide, the pertussis, Haemophilus influenza, heavily invested in influenza virus research, typhoid vaccines, the neuraminidase inhibitors, and other contributions to the sort of the medical education and research here at Baylor. So in a sense, that side project, I was kind of lucky that I was participating in it, because soon, you know, when the NIH funded TB project finished, I fully joined the group, which is heavily sort of centered around the vaccine and treatment evaluation unit. This is a multi-decade project that is funded by the NIH, initially started by Dr. Couch, who passed the baton to Dr. Keitel. And this particular NIH-funded, used to be a contract, is heavily invested in vaccine research against respiratory and gastrointestinal pathogens, but their priority was the rapid research response to public health emergencies. Hence the COVID link there that has been ongoing for decades. And before COVID, there was the big yearly epidemic that we see here with occasional pandemic, and that is influenza virus. The Baylor VTEU has been involved in research pertaining to vaccine development, evaluation of epidemiology, evaluation of therapeutics, and evaluation of diagnostics pertaining to influenza throughout all these years. I joined somewhere here when H5 virus, the avian influenza, became an issue. So during those following years, I worked on clinical vaccine development as it pertains to H5 N1, H7N9, the 2009 pandemic, H1N1, what became a sort of a surge in activity here at the center, as well as others. So you can tell from the slides that the issue of being sort of groomed or prepared to answer to the COVID-19 pandemic began somewhere in the 1960s here at the center. And there's the other component that sort of parallels this the influenza, which is the center here, or the all VTUs are also engaged in response to potentially bioweapon agents. And actually my very first project was in clinical vaccine research pertaining to Francisella tularensis, which is a disease that causes severe disease here in the US, with a variety that causes a mild illness in Europe and Central Asia, but it is a category A pathogen. In 2016, I became the VTU PI. In 2019, submitted the renewal with the collaboration of the center. And on the 21st of December, we got the notice of award of the VTU grant, only to have on the 30th of December 2019, so that's less than 10 days later, these emails pertaining to a curious pneumonia occurring in China that is causing significant morbidity and mortality. That was the SARS-CoV-2, I don't need to tell you that, which as of yesterday has claimed the lives of more than 4.6 million individuals all over the world, with 226 million cases reported. There's probably a significant underreporting of cases there. And in the US, continues to wreak havoc in terms of hospitalization, death, and societal disruption. Right out the gate, we had to be ready, and we started with the Adaptive COVID-19 Treatment Trial Act, which had 4 iterations: Act 1, 2, 3, and 4. Act 1 and 2 results are published, 3 and 4, one about to be published, one under construction still. Regardless, each one of those studies enrolled 1,000 individuals. At Baylor, we enrolled patients at Baylor St. Luke's and at Bentob, and contributed sort of our patient population and national expertise on the weekly sort of protocol meetings. Act 1 evaluated remdesivir versus placebo. It was a randomized, double-blind, placebo-controlled trial, enrolled a little over 1,000 COVID-19 patients who were hospitalized. The randomization was 1-to-1, so 541 received remdesivir, 521 received placebo. The primary endpoint was the recovery, the time to recovery, and the study demonstrated that the median recovery time was decreased from 15 to 10 days with the addition of placebo, with the addition of remdesivir to the treatment of these patients. And that was statistically significant to the point where the DSMB requested towards the end of the study, when these data were shown to them, to unblind, and if someone is still hospitalized, to offer them remdesivir. The 28-day mortality on the study was 11.4 in those who received remdesivir versus 15% in those who were on placebo, which is a 27% reduction. The study was not powered to answer the question of mortality, but at a minimum, the number was in the right direction. The study was also analyzed by the baseline ordinal scale, which is going from 4 are patients who are hospitalized with COVID but not needing supplemental oxygen to those needing supplemental oxygen in ordinal scale 5. Ordinal scale 6 is non-invasive ventilation or high flow oxygen, and ordinal scale 7 are the mechanically ventilated or ECMO patients. You can see that the intervention favored remdesivir it favored it as a group, but when broken down by the ordinal scale, the effect was most pronounced in those requiring oxygen but not high flow oxygen. Just recently published is another post hoc analysis of Act 1, where we evaluated the progression to mechanical ventilation or death, when this actually kind of a popular way or meaningful way of evaluating the efficacy of certain interventions in COVID. Remdesivir was associated with the reduced risk of progression to mechanical ventilation or death, with a hazard ratio of 0.67 that was statistically significant. But the Solidarity trial, which is a large international trial that evaluated remdesivir and other therapies in patients hospitalized with COVID. However, in that trial, which is called randomized, but the randomization is to what's available in a particular hospital and to what is accepted by the treating physicians. And those statistically minded amongst you can potentially argue that that's not a randomization, and potentially they are correct. It was open label, with patients having the ability to cross over. There were many issues raised with its appropriate classification of patients, the quality of care, and the potential biases introduced. Nonetheless, these were the most important findings when it comes to remdesivir. There was no difference between remdesivir and control, with the primary endpoint being in-hospital mortality. Remdesivir right now is approved for the treatment of patients with COVID. Most treatment guidelines will include it in their recommendations for hospitalized COVID-19, although some are excluding mechanically ventilated patients from receiving or being recommended remdesivir based on the subgroup analysis, which I just showed you. And hospital-based committees still continue to recommend it though. So there's a lot of variability there. Since then, multiple effectiveness analyses have demonstrated mortality and clinical benefits of remdesivir in all hospitalized COVID-19 patients. Then we had to, we barely closed Act 1 to open Act 2. The inflammatory cascade in COVID-19 is probably one of the biggest drivers of severe disease and mortality in these patients. So baricitinib, which has both antiviral properties, but importantly is a JAK inhibitor that is relatively safe, and it prevents a lot of the inflammatory the expression of the inflammatory markers that have been implicated in that inflammatory cascade. We followed the same design. It was randomized, double-blinded, placebo-controlled, where half the patient received remdesivir and bari and half the patient received remdesivir and placebo. The population is the same, hospitalized patients with COVID, pneumonia, and the outcome was the same. or the primary outcome, which is time to recovery. The study enrolled 1,033 patients, and 20% of them were mechanically ventilated. The study did also show that adding baricitinib to a background of remdesivir did decrease the median time to recovery. However, it went from 8 days to 7 days. That was statistically significant. The other important finding is the 30%, there was a 30% higher odds of improvement in the clinical status on day 15. And that was also clinically statistically significant. The 28-day mortality was 5.1% in the Bari group versus 7.8% in the remdesivir group. It was not statistically significant, but then again, that was neither a primary outcome nor a key secondary outcome for which the study was powered. But again, the numbers are in the right direction. And the issue of the modest clinical benefit of baricitinib was brought to the fore, given the reduction in time from 8 to 7 days. But there was a follow-up trial that was not NIH funded, and that just got published, but it was high quality in that it was randomized, placebo-controlled, double-blind, large clinical trial where 1,500 patients were randomized. It did exclude mechanically ventilated patients, although a follow-up study with the same design was performed, and the data of that other one will be soon forthcoming. The primary endpoint was progression to ventilation or death, and the key secondary outcome was mortality. For the primary outcome, there was a reduction in the progression to mechanical ventilation or death when Bari was used compared to placebo, but it was not statistically significant. However, the key outcome of mortality, for which the study was powered, did show a significant mortality benefit for baricitinib. And that trial was performed at a time when steroid use was high among patients with hospitalized COVID-19. And the effect was seen in both patients on steroids and those who were not on steroids. You can see here that the difference even actually was more pronounced in those who were not on steroids. Baricitinib did receive EUA for adult and pediatric patients hospitalized with COVID-19 and requiring supplemental oxygen, mechanically ventilated or ECMO. That's the FDA EUA based on the clinical trial findings. But the clinical societies that did incorporate baricitinib, most of them did in their treatment guidelines, focused it on the ordinal scale 6, which is where the most pronounced difference was seen. So why is there a difference between the high quality clinical trial inclusion criteria and then how the treatment guidelines for both remdesivir and baricitinib have evolved? And that leads me to another important lesson I learned in clinical research over the years, is that really the most important section of any clinical research project or paper to be able to interpret the results are the methods. It's the section that we all glance over and we don't want to read because it's written in a way that is so dense and so not reader friendly, but in it is the key to interpreting the findings. And I learned that the hard way because another mentor, Dr. Greenberg, tapped me to participate in the practical reviews of infectious diseases, maybe 10 years ago now, where I had to review and interpret the results of 2 clinical research publications per month to a national audience. And of course, I picked the 2 articles based on reading the abstracts. And when the deadline comes to submit the article, I read so I can, write up the summary and the interpretation, only to realize that the abstract really does not match what the methods and results in the papers are showing. And if I can impart one lesson, is that happens way too commonly, even in journals that are high impact. So I learned that the hard way, because I would invariably pick articles that end up being not as meaningful as they claim to be. And since then, I also, continued to work in reviewing and interpreting clinical research paper for another entity here, the New England Journal Journal Watch. But again, invariably reading the methods of how anyone gets to their results is the most important part of the paper. Moving on to COVID-19 vaccines. So in April 2020, actually move back, maybe January 2020, the phase 1 for the mRNA vaccine was to be conducted within the VTU. I submitted that we would like to do it here at Baylor. I got declined. However, in April 2020, I was asked to lead the phase 3 clinical trial. And to do that, we had to learn, or at least read and understand certain lessons about coronaviruses. There's the seasonal corona, there's the epidemic corona, which is in the form of SARS-CoV-1 and MERS. But really, there was some interesting literature on seasonal coronavirus that was done in the 60s, 70s, and the last really interesting challenge took place in 1990, I think, or 89. So what do we learn from these previous old important publications? Here's a study where the young healthy volunteers were inoculated with seasonal coronaviruses. We see that 50% of those who are probably multiple times previously exposed to coronaviruses, you can infect half of them. And the overwhelming majority will be mild to moderate disease, as you can see here. Another important findings that got replicated in some of the other papers is that despite repeated exposures to seasonal coronaviruses, the neutralizing antibody titers will wane. As we can see here, in 8 out of 24, they had a titer less than 5, which is, I presume, is the cutoff here. And only 4 out of 24, 4 out of 26, I'm sorry, 4 out of 26 will have really good titers, good neutralizing antibody titers in the serum, despite multiple repeated exposures in the past. However, there's a trend in that those with a higher neutralizing antibody titers are less likely to get infected, to develop a cold, than those with lower antibody titers, but nonetheless, previous infections are partially protective against disease. They are not protective against reinfection, and neutralizing antibodies wane over time. So we set out to design that study, which actually ended up being sort of the standard, or not standard, the first one to get designed, and because all the phase 3 had to be harmonized, it sort of set the tone of what followed. So we could, what would be the primary endpoint of the study? COVID tells us that there is a lot of infection, little less disease, and much less severe disease. If we were to have the primary study endpoint as infection, that would have required frequent sampling, test validation, which at the time was not even, it was still happening, it was not there. And we need to keep in mind that many successful vaccines prevent disease, but not necessarily infection. Then there is the endpoint of disease, which is symptomatic. driven by symptoms and can be virologically confirmed with PCR, and that's a feasible endpoint. And then there is severe disease, which would have required a large sample size, thus impacting feasibility if we are to get an answer in a short amount of time. So a study with a primary endpoint that captures all of these endpoints is likely not feasible. The disease seemed to have been here sort of the Goldilocks answer. Oh, but what is the design? The design could be the individual randomized clinical trial in which there is randomization at the subject level to the vaccine or placebo. It is a statistically efficient method and a value when there is heterogeneity in disease incidence. Another option would have been the multi-arm individual randomized clinical trial. And in that one, you can, if there are multiple candidates, you can randomize to multiple active interventions and one placebo. It's quite efficient and can diversify the portfolio of candidates reaching the market in a quick time and minimize the effect of temporal trends on disease epidemiology. There's the parallel cluster randomized clinical trial where the randomization occurs at the level of the unit, whether it be the household, the community, the ring network randomization. It allows a measurement of the total direct and indirect effect of the vaccine, but it's less efficient to conduct than an individual randomized clinical trial. And then there is the stepped wedge cluster randomized clinical trial, which was used in the evaluation of vaccines against Ebola and the epidemic of Ebola a few years ago. The vaccine is given to all subjects in a randomized order, meaning some people get it early, some people get it late, but they are randomized to the time when they get the vaccine to eliminate confounding. It is rather complex to plan and analyze and slow to perform. So at the time, there were multiple constructs were moving into phase 3 clinical trials sometime in the May 2020 timeframe. So arguably a multi-arm randomized clinical trial would have been the most efficient way to test for the vaccine efficacy of these constructs. However, it would require collaboration and planning between various pharmaceuticals, which is difficult. And there was variability in the readiness of some of the constructs to launch within a similar time frame. So it landed on the individual randomized clinical trial. And sort of all of the phase 3 clinical trials evaluating the COVID-19 vaccines followed quasi-similar primary endpoint randomization schemes, and they launched one after the other. The Moderna and Pfizer one were leading the pack, and they both launched in the last week of July. The launching and the implementation were not without their drama. One important clinical trial principle is that the study population has to mirror the vaccine target population. And if you, one important problem is that COVID-19 was disproportionately affecting minorities in terms of incidence and mortality, but clinical trial participation is historically predominantly affluent Caucasians. So there was this disconnect. And of course, we wouldn't want to develop a vaccine only for it not to be implemented in certain population just because of underrepresentation. And that took a toll on the study, actually. Here in red is enrollment numbers of Caucasians, of whites on the study. And in blue are the non-Caucasians. And you can see, as with all other clinical trials, the Caucasian enrollment was rather robust. And despite, from day one, sort of aiming to have a representation of a minimum, the country. But an intervention had to be done here when it was evident that we were failing at doing that in the beginning. A directive was sent to all 99 sites to halt enrollment of Caucasians, except if someone's already on the books, and to sort of encourage and accelerate the enrollment of non-Caucasians, non-whites on the study. Eventually, that led to the study taking longer to implement, but it achieved its goal. For example, the Moderna clinical trial had 20% Hispanics, 10% African American, 4.5% Asians, and 2.1 and were multiracial and other. So rather a representation that is attempting to be close to the country demographics. The vaccine was safe. There were systemic adverse events that we noted on the study. Any adverse event, any systemic adverse event is represented here, but you can see between dose 1 and 2, there's an uptick in grade 3 systemic adverse event from 3% to 16%, mainly headaches, fever, fatigue, and myalgias. All of it almost resolved within the first few days. Importantly, the study did find that the vaccine was efficacious. The vaccine was 94% efficacious at preventing symptomatic COVID. And this is where it paid off to have the representation of elders, people at risk, people of racial and ethnic groups, because the vaccine could demonstrate that the efficacy is maintained in all these subgroups, with the numbers being very similar to the overall efficacy of the vaccine. The findings that took place in the Moderna clinical trial were replicated or were very similar, even down to the numbers, with the Pfizer mRNA vaccine phase 3 clinical trial. But the other vaccine that was also eventually that reached the market via EUA closely thereafter was the adenovirus 26 based vaccine. That's the Janssen product, Ad26cov2.S, which also was government funded, international randomized placebo-controlled study. Individuals received 5 × 10⁻10 viral particles per milliliter in one dose, and it was intramuscularly. And the primary endpoint was moderate to severe critical COVID-19. But if you read the methods, you will realize that it was pretty much the same endpoint in all phase 3 clinical trials, which is symptomatic COVID. And this was corrected in the FDA briefing, but didn't make it really to the publication. The Janssen study was also very successful at enrolling individuals who belong to racial and ethnic minority. 45% were Latinx, and Black or African Americans were 18%. The study had sites in Latin America and in South Africa, thus contributing to the diversity of these participants. The Janssen product was less reactogenic than the Pfizer or Moderna ones in terms of systemic reaction, or you can see here that 0.6% of these systemic reactions were grade 3 compared to somewhat 3% or 16% in the Moderna and Pfizer one being grade 3. The vaccine efficacy of the Janssen was measured in 2 ways as a primary endpoint, starting at 14 days or starting at 28 days. But they were comparable when it's preventing all COVID, 66.1 to 66.9%. The vaccine efficacy was higher for the prevention of severe COVID, 76% starting 14 days and 85% starting at 28 days. All three vaccines received their emergency use authorization. And post EUA, there was further refinement of the safety findings because we followed volunteers only for 2 months as a median before submitting for EUA. In the Janssen Ad26 vaccine, there was in the trial imbalance in thromboembolic event, 15 versus 10. Post-marketing, there was, we identified, or actually in the US, via the CDC and via European safety surveillance, the syndrome of vaccine-induced thrombosis with thrombocytopenia, which resembles clinically a lot the syndrome of heparin-induced thrombocytopenia with extensive thrombosis, including arterial, but somehow it had a predilection for cerebral veins. It occurred in 1 in 100,000, and it did occur with both the Ad26 product and in Europe, the AstraZeneca product, which is a chimp ad-based vaccine. Another post-marketing safety signal was detected in terms of Guillain-Barré, 1 in 100,000 as well. For the mRNA vaccine clinical trial, there was imbalance in the Bell's palsy occurrence. On the Moderna, it was 3 versus 1. On the Pfizer, it was 4 versus 0, I think. But post-marketing, there was the Bell's palsy signal fell out, and it was basically the incidence was comparable to background. However, there was anaphylaxis that was detected in the post-marketing surveillance at an incidence of 2.5 to 4.7 per million, and myocarditis pericarditis occurring in 12 per million. However, it's very age and sex dependent, meaning it occurs more commonly in the younger, with the highest incidence being in the adolescents, and more so in males than females. It doesn't resemble quite the run-of-the-mill myocarditis we see, in that it resolves quickly, and I think the median duration of hospital admission is 1 day, but nonetheless, it is under extensive scrutiny right now in terms of detecting and describing these cases. And then after all the work we do on a clinical trial, the important thing is the real world effectiveness. Initially, after the mRNA vaccines were marketed, and I say mRNA because they sort of encompass 90% of all the vaccines we got here in the US. So most of the data is really driven by these two vaccines. Although in the last MMWR, we are starting to see Janssen representation. Regardless, post-marketing, the effectiveness of these vaccines mirrored almost to the number the efficacy observed on these clinical trials. But that was in the pre-Delta era. So that was the Alpha era here in blue. So against any infection, US, Scotland, and Qatar data show that these vaccines are in the 90% effective range. Symptomatic infection, same thing. Hospitalization, close to 95% to 100. And severe disease, here is driven by one paper, it's up to 100. Then the Delta reared its head and became the predominant strain causing disease pretty much everywhere in the world. And we saw a decline in the real-world effectiveness of these vaccines. And it all depends, and the magnitude of this decrease depends on the paper you read, but it's mostly against these first two columns, any infection or symptomatic infection, depending on the method of the paper. We can see here that it dropped to the 80s in the data from Scotland and the US, and somehow in Canada, it's not seeing a drop, but definitely the confidence interval got much wider. And in England, in Qatar, it dropped to the 50% range. However, when we look at hospitalization, and severe disease, and I don't have a slide here for death, the real-world effectiveness of this vaccine to this day remains maintained with little to no waning. The only potential signal at waning I saw only in one MMWR release, and it's in individuals over 75. But all in all, it remains a success. And I know now there's an epi group in Baltimore that is trying sort of to contribute to the discussion of the vaccine effectiveness against Delta. Is what would have happened had we not had these vaccines in the market in the US. To sum up the real world effectiveness and vaccine efficacy only in one slide, we see here that if we evaluate all the reports taken together, the efficacy against any infection, even if it dwindles, depending on the strain, the methodology, the vaccine under evaluation, the efficacy against severe disease is maintained. And that remains true with the alpha, delta, and beta. The gamma drops down in terms of effectiveness against severe disease, but remains at the 80% plus. When we evaluate it by construct, and these are the two constructs in the US, you can see that even when the efficacy drops against the any infection, it does not drop much against severe disease. And here are some emerging data on early versus late, even when for those vaccinated later, the efficacy against any infection will drop, but not the efficacy against severe disease. Which leads me to answering now the questions that Rolando wanted me to answer. The two red questions are the ones I'm going to attempt to answer first. What led you to become a physician scientist, and what stimulates you to remain as a physician scientist? I tried to think closely on how to answer these, but I realized that Rolando is asking the wrong question, because it's really who led you to become a physician scientist and what stimulates you to remain a physician scientist. The what is at times less important. So we have here Dr. Wendy Keitel, probably the sharpest tool in anyone's box, who is a fearless leader here at the center and who mentored me. Dr. Couch and Dr. Gleason, while I didn't work with them on projects, they sort of cast a shadow over a lot of the things we do around here in terms of setting standards of excellence, standards of approach to certain problems. Dr. Atmar, who, in addition to many of his accolades, probably has the most perspective, and I needed that in the last year and a half. Jenny, Erin, and Christine, the young talent that made a lot of the work possible while I was being drawn to all sorts of teleconferences around the clock, and who will one day hold the baton. Tony, with his ever positive energy and putting sort of the lab perspective in a lot of what we do. And Erin, also one of our young physicians who is now taking more and more role in pediatric clinical trials along with Mary Healy Dr. Dan Musher, I'm reminded that he asked me, actually, he hired me, and then, he was the section chief, and he asked me a year or two into the fellowship. So are you going to be in the research field or clinician? And I adamantly answered, no, I'm going to be in the clinician track. And he did this to me. We'll talk about it later. And as I was preparing the slide, I realized something that completely crossed my mind, but as I was putting them together, I remember telling him, oh, I'm going to work on the TB project. He said, well, why don't you work on a project with the people in the vaccine center? And I said, no, I want to work on TB. So now I'm reminded, that he told me from the get-go, you probably should go there. So thank you, Dan. And then the other question is, how does your clinical practice influence your research, and how does your research influence your practice? We bring questions from our clinical practice to influence the research. I can think of the last proposal that we submitted, which is to improve the treatment of syphilis, for example. How does research influence my practice? I learned after all these years of reading, writing, interpreting papers, that a lot of the certainty we put in things is just not there. And to take things, within limits when it comes to certainty of what we are doing to our patients. And with that, I'm happy to answer questions.
Thank you so much, Dr. El-Sally. That was outstanding.
Thank you.
Thank you for correcting my my question that I'm asking the wrong question.
At least for me, I don't know for the others.
There were two aspects of your talk that I liked everything of your talk. It was fantastic, but there was two that stood out to me. One is the focus on the methods, but the second one that I really, really liked is that you started off by looking up at articles published in the 1960s. So going back to what has been done already, and realize that a lot of research has been done in the last millennium, and we can often get a very valuable information from those as we design our studies. So the audience can put questions in the chat. I'm going to ask just a practical question on these clinical trials for vaccines. So in the Moderna trial, for example, and once you have reached the point that demonstrate the effectiveness and there is an emergency use authorization, what do you do from a practical standpoint to the participants in your trial, especially those in the placebo group? Can you comment on that?
Yes. So we had to redesign the trial, and we amended the protocol to unblind the participants and offer the vaccine to the placebo recipient. We had to do that because we were talking about a direct morbidity and mortality benefit. Well, direct morbidity benefit. So everyone on the trial got the vaccine. And after that, as you can imagine, the study is no longer a placebo-controlled randomized trial. It became more of an observational cohort, if you will.
Great, thank you. A question from Dr. Shaw. These mRNA vaccines have amazing efficacy. With this new technology, do you foresee this technology towards other viruses like RSV or even other uses?
Yes, that is a field now of intense research. They were already in the pipeline, mRNA vaccines against Zika, human metapneumovirus, CMV. They were in various stages of development. I can see that the success against one pathogen is probably going to invigorate the pipeline that way against other pathogens, but that's not to say it's going to be successful. So just because they were successful against one pathogen, it doesn't mean that they will be against others. And but we will see. I know that they are beginning developing products against other emerging pathogens that are on the list of high priority pathogens like the Nipah, the HIV vaccine. I think there's a construct that's about to enter or already entered into clinical research. So it did invigorate the field, and we will see if it works.
Okay, thank you. There's a question from Dr. Kulkarni. It's somewhat related to my question of unblinding, but I'll read it. A report came out in New England Journal of Medicine describing long-term safety and efficacy of the Pfizer vaccine after 6 months. However, as we discussed, once the EUA became available, participants are unblinded and patients who got placebo get the vaccine. So how is long-term vaccine efficacy evaluated now in this type of situation?
So the efficacy, we have data up until 6 months. So as you know, we published after a median follow-up of 2 months, and the EUA was submitted based on these data. But as the wheels were in motion, subjects were still blinded until we got the EUA. So we have, for both Pfizer and Moderna, blinded data up to 6 months. Just today, the data for the Pfizer came out, and ours is coming next week. It will be published. So we will have safety and efficacy in a blinded placebo-controlled way soon, with the 6 months follow-up. Now, after that, it's no longer blinded. It's closer to effectiveness than efficacy. And that is why the data we are seeing in the real world are very important to gather and to analyze and interpret. Of course, there are the quality of data in certain places are better than others. For example, in places where there's a national health system where the data of everyone is linked centrally, and they are used to cranking these data on a regular basis, say for other pathogens, they're probably ahead of the game. But the CDC is also conducting its own rigorous data on real world effectiveness. And we are seeing them put these data on Fridays, one after the other in the last few weeks.
Okay, thank you. A question from Dr. McGinn. Amazing talk and work, very thoughtful. Agree, methods, the most important aspect of a publication. One question, I was surprised by the perceived mortality benefit of remdesivir. It did not meet statistical significance, but the magnitude is more than I thought and seems inconsistent with clinical experience.
I am not, the mortality with remdesivir was reduced, but not statistically significant. I hope I was clear on that one.
Right.
In the clinical trial. However, when you analyze disease progression to mechanical ventilation or mortality, which is another way of assessing the data, and then that's a post hoc analysis, it was statistically significant. But you're right, looking at mortality alone, it was reduced, but the p-value was not statistically significant.
Thank you. And well, actually, we're just about at the end of the talk. Dr. Shah, any final comments?
No, I want to thank both of you for a terrific rounds, very timely and great information. Thank you.
Great. Thank you very much. And thank you to the audience for participating.
Thank you for inviting me.